Hi everyone, my name is Savya. I'm currently a sophomore at Dublin High School. I'm honored to present my research on optimizing and stabilizing induced pluripotent stem cells processing efficiency. I conducted this research under the mentorship of my mentor, Ms. Lana Ho, and today I'll be discussing the current challenges that iPSC generation faces, as well as highlighting a couple of novel techniques that may enhance their viability in clinical settings. Okay. Stem cells have revolutionized regenerative medicine. iPSCs in particular allow us to generate patient-specific cells for therapies while avoiding ethical concerns associated with embryonic stem cells. However, challenges such as low reprogramming efficiency, genetic instability, and immunogenicity hinder this widespread clinical use, as well as many research applications. My research hopes to overcome some of these limitations and prevent and propose novel techniques to make this more viable and widespread for use. Okay, so what are iPSCs? Induced pluripotent stem cells, also known as iPSCs, are generated by reprogramming adult stemmatic cells into a pluripotent state. Dr. Shinya Yamanaka identified four key transcription factors in 2006, those being OTC4, SOX2, KLF4, and CMYC, that induce this transformation. iPSCs can differentiate into any cell type, making them highly valuable for regenerative medicine, disease modeling, and new drug discoveries. So how are iPSCs currently used in both a clinical and research setting today? In clinical settings, they're used to treat Parkinson's disease. iPSC-derived dopaminergic neurons replace lost brain cells. In diabetes, they can be used to reprogram pancreatic beta cells that produce insulin. In cardiovascular disease, they create cardiomyocytes for heart tissue regeneration. In ophthalmology, they help develop retinal pigment epithelial cells to restore vision. This was one of the main use cases that Dr. Yamanaka highlighted in his presentation. As far as research applications goes, iPSCs can be used for disease modeling, high throughput output screening for pharmaceutical testing, as well as personalized medicine and tissue reengineering. What are some current limitations of iPSCs? One of the main limitations stopping this from being a widespread method of treating diseases is their low reprogramming rate. Only about 1% of adult somatic cells are able to successfully reprogram into iPSC cells. They have high risks of genetic instability due to some of the transcription factors used, which can increase risk of malignant tumor formations. They have high risk of immune rejection, even though they might be derived from the patient receiving the treatment, as well as several maturity issues which stop iPSCs from fully maturing and developing their functionality, which again ties back to only 1% of stem cells being able to be used fully. Before I go further, I just wanted to highlight how how iPSC generation is currently addressed. The process of iPSC generation begins with stemmatic cell isolation, where fibroblasts from the skin or peripheral blood cells are collected through biopsies. The extracellular matrix is broken down using enzymes such as collagen releasing fibroblasts, which are isolated via centrifugation. Next, in the cellular reprogramming stage, transcription factors such as OTC4, CMYC, KLF4, and SOX2 are introduced to the host genome either using viral or non-viral vectors, with the latter being more popular currently. Cells are then cultured in a reprogramming medium such as Essential 8 or MTESR1, supplemented with growth factors such as BFGF to support growth. The culture is maintained on feeder layers such as irridated mouse embryonic fibroblasts or extracellular matrix coatings such as mitrogel or laminin. A hypox environment is maintained to enhance reprogramming efficiency. Over the next 2 to 3 weeks, these colonies undergo transformation. During this phase, they develop higher nuclear to cytoplasmic ratios, mimicking properties sort of like embryonic stem cells. Colonies that show a unique or uniform formation are then selected to be expanded. Once these colonies are established, they're tested for pluripotency, where the cells are tested for key markers, including OTC4, SOX2, NANOG using immunostaining or qPCR methods. Finally, the application and banking stage. iPSCs are derived into various cell types, including ectoderm, mesoderm, endoderm, and germ cells for research or therapeutic purposes. Alternatively, they can be cryopreserved for future use, ensuring stability and a stable supply of reprogrammed cells. So what are some new emerging ideas that aim to address some of the shortfalls of iPSCs? Chemical reprogramming. Instead of using the traditional Yamanaka factors, small molecules can be used to induce pluripotency, reducing genomic instability and increasing efficiency. mRNA-based reprogramming. In transit mRNA transfections allow for reprogramming without DNA integration, lowering the risk of mutagenesis and improving clinical safety. CRISPR-9 and Cas9 applications. Gene editing can enhance iPSC stability by correcting mutations and reprogramming state in the reprogramming stage, improving consistency and reducing the risks associated with tumorigenesis. Um, Strategies for enhancing iPSC safety. The risk of genetic instability and tumorigenesis remains a major barrier to clinical use and research. The researchers are implementing following methods: non-integrative reprogramming methods, viral vectors like the Sendai virus, and non-viral methods such as episomal plasmids prevent permanent genomic alterations, reducing the risk of mutations. Epigenetic modulation by targeting histone modifications and DNA methylation patterns. Researchers can improve the stability of iPSCs, ensuring more reliable differentiation and functionality. Alternative programs, some efforts focus on replacing CMYC, which is the main component in the pluripotency of these cells with safer alternatives such as NONOG3 and LIN28, reducing tumorigenic potential while maintaining reprogramming efficiency. Improving functional maturity. Therapeutically viable iPSCs must mature into fully functional cells. Several approaches are being explored to enhance differentiation. 3D culture systems. Traditional 2D cultures fail to mimic the complexity of in vivo conditions. Using biomimetic scaffolds in 3D cultures provides a more realistic environment for cell growth. Co-culture with mature cell types. When exploring specific tissues, combining iPSCs with potential differentiated cells such as neurons with astrocytes can improve cellular interactions and tissue development. Signaling pathway activation. Modulation pathways such as WANT and Notch enhance differentiation by guiding iPSCs towards specific cell fates with improved functionality. Large-scale iPSC production. To meet clinical demands, iPSC production must be scalable and consistent. The following advancements are addressing these needs. Bioreactor systems. Automated high-volume cultivation of iPSC ensures reproducibility and standardization. AI and machine learning. These tools analyze cell morphology and gene expression and look for higher quality iPSC lines, improving efficiency and reducing variability. iPSC biobankings. New developments in biobankings allow iPSC lines to be stored for future applications, which reduces the need of going through this process over and over. So what lies ahead? CRISPR-9 and CRISPR-enhanced iPSC therapies. Gene editing techniques will enable the correction of disease-associated mutations before transplantation, opening doors to personalized medicine. Regulatory guidelines, standardized protocols, and clinical regulations will be essential to transition iPSC-derived therapies into widespread medical use. In addition to this, antibody transcription factor conjugates is a form of foregoing genetic instability. This is a completely novel idea that I had published at the Journal of High School Sciences. I'll be further researching this, and I hope to present once again at the Polygence Symposium regarding my findings after the summer. That is the end of my presentation. Before I wrap up, I'd just like to thank my mentor, Ms. Lana Ho, the Dublin High School Biomed Academy for their support, and all of you all for your attention. And I'm open to take questions.
Amazing. Thank you so much. One question that I just had was, at the beginning of the presentation, you mentioned because of some of the challenges that we're missing out on like even more, like novel and improved techniques. I'm curious to like hear what are some of those, opportunities that we aren't quite there yet, but that you think are like kind of the most exciting, right? Like if this kind of becomes more perfected, what are you most excited about that opening up?
Yeah, so I just quickly switched over to the slide I
Yeah,
think you were
thank you.
mentioning. So one of the main like drawbacks that this thing that IPSCs currently have is their low reprogramming rate, around 1%. It's not viable for wide scale use as you can't scale it up. Once we're able to say increase this to around a 75% reprogramming rate where it becomes much more scalable and safe for clinical use, we'll be able to generate new tissues. And I mentioned drug development. Currently we test vaccines on live patients. This would allow us to look at how drugs, vaccines would react with patients using real human tissue without actually having to test it on a living patient.
Wow, so neat. Does that mean too that it would reduce the need for animal testing as well?
Yes,
Hmm.
it would. There are a couple of drawbacks to that. Um, Currently, this isn't really giving life. It's more so creating living human tissues. So we m- more probably examine like how this would affect human tissues and cell types. As far as animal testing goes, we would still need to see adverse effects on living creatures as this would not completely show like the full extent to the drug's effects or side effects. But it would definitely be addition to drug development and help speed up processes in vaccine development as well.
Very cool. My last question for you. You mentioned your mentor, Lana. Um, what was something that she like helped you kind of best understand, or maybe like kind of a topic or idea that she kind of introduced you to that, you'll always kind of remember working with her for.
Yeah, so this diagram that I have, my mentor really helped me make this.
Oh,
It's
cool.
completely self-made, and one of the main struggles that I had like comprehending this was the different methods in which iPSCs or the transcription factors are introduced to iPSCs. I really had trouble distinguishing between viral and non-viral vectors, and I feel like her knowledge on the field really helped me fully grasp the concept to its fullest. And yeah, that's one of the main things. She also really helped me structure my paper. It was my first paper coming out of around sophomore year. So it was like really fun just working with her. And I really look forward to have like the chance to work with her again.
Yeah, amazing. Well, thank you so much both for sharing kind of your experience, and also your findings in this amazing presentation.